Good day, classmates. This is a report about viroids and prions. This report will cover the following topics: the general characteristics of viroids and prions, their taxonomic classifications, species update under viroids and prions, and finally, we also added a topic regarding how you treat viroids and prions.
We will first describe both viroids and prions. Now, starting with the viroids, these are single-stranded RNAs with a capacity to self-replicate. Viroids always look circular, or, if ever, an amalgamation of circular RNAs that can form distinct shapes, which is usually seen in like a straight line. But unlike its cousin, or rather its distant cousin, the virus, viroids do not produce any kind of protein. This incapability is the reason why these RNAs do not have any protein casing at all, to which they lack protection compared to the virus's capsid. Since viroids can self-cleave, they do not need to attach to a host cell to reproduce, making them a very good pathogen in an organism. Successful as this process may be, most of them all are only capable of infecting plants, although some studies say that there is a viroid-like disease that is spread through humans, which is the hepatitis D. Now we go to the prions. Generally, prions are proteinaceous molecules that are erroneously shaped in the form of beta sheets. These infectious pathogens are actually smaller than viruses, and while they are in itself proteinaceous, they do not contain any genetic code, no DNA or RNA at all. That gives the question, how do they infect other organisms and how do they reproduce? In this case, the beta sheets formed by these prions actually induces other normally shaped proteins, for example, an alpha helical protein, to convert their conformation into beta sheets. Moreover, this pathogen often targets encephalopathic systems like the brain and neurons, which is why prions are regarded as dangerous. There are no cures for prion diseases. The reason being is that these misfolded proteins are actually tough and can only be denatured at a very high temperature. For example, up to almost 900 degrees Fahrenheit to prevent misfolding. No human can withstand those temperatures, which makes these pathogens very lethal.
Prion proteins and the prion infectious cycle. So the following are the prion proteins. The first one is the prion protein PRNP, which is existing in the host cell. The second one is the prion protein cellular PRPC, which is the native form of prion that is encoded by the prion protein PRNP. PRPC is situated in the neurons of healthy animals, especially in the brain. The third one is the prion protein scrappy, which is the pathogenic form of the prion protein. The prion protein cellular and the prion protein scrappy have the same amino acid sequence but in different configurations, given that it is from the same animal species. Infectious cycle begins when the PRPSC enters the host cell, initiating the conversion of PRPC into pathogenic form. Afterward, the pathogenic form of prion will multiply by transforming the pre-existing native prions into pathogenic forms. Insoluble crystalline fibers or amyloids in the neural cell are formed when the pathogenic prions accumulate and amalgamate. This will result in the manifestation of the symptoms of the disease related to brain and nervous tissues. The membrane attachment of PRPC is needed before the disease symptoms start to show.
Now let's discuss the classifications of viroids and prions. The schematic taxonomical classifications of viroids was first proposed in the early 1990s, and at the time, there were only 27 species identified. Also, the taxonomy of viroids, as illustrated in the picture, does not stretch beyond the category of family, since their evolutionary lineage have not yet been fully examined even up to this present time. Back then, it was also proposed that gene sequence data is the most viable criterion for identifying viroid species and that only 1 to 10% error is allowable to determine whether the viroid examined is the same species as the viroid it was compared with. In the year 2014, however, there were updates regarding newfound species in the genera Popsiviroid and Abscaviroid. Additionally, there is a new genus established, the Elaviroid, containing its sole species as of now, which is the eggplant latent viroid. As for the taxonomic classification of prions, quoting from Fenner's Veterinary Virology by McLachlan, it was stated that prions have not been classified the same way as viruses or even viroids, which have your families, genera, and species. However, prions are identified by their host species, named after the clinical disease that they cause, and their associated lesions or injuries that their infected hosts experience.
Here are some representative species under viroids and prions. Under viroids is the potato spindle tuber viroid that infects potatoes, causing stunting in the plant as well as malformation and cracking of tubers. The tomato planta macho viroid that causes the loss of chlorophyll in disfigured and brittle leaves, which also leads to having very small tomatoes. The avocado sunblotch viroid, which is the smallest viroid discovered yet, can cause sunken, yellow to purplish longitudinal scars or broad spots on the surface of avocado fruits. And the peach lane mosaic viroid that causes necrosis of flower buds and branches, as well as wounding of ripened fruits. An example of a representative species under prions is PRPSC or scrappy associated prion protein. PRPSC is the disease-causing form of cellular prion protein or PRPC. It arises spontaneously in the brain tissue and affects the brain, causing transmissible spongiform encephalopathy or TSE diseases. One type of this is the variant Creutzfeldt-Jakob disease that is transmitted by eating contaminated cattle products and by secondary blood-borne transmission. Another is Kuru that is transmitted through eating infected meat. Species update on viroids. Viroids are commonly found to infect mainly dicot species only. However, a paper from the group of Yang in 2020 states the discovery of a new monocot infecting viroid in Dendrobium TSA. It is temporarily known as Dendrobium viroid or DVD, and its ineffectivity and circularity were proven in the officinal. It is predicted to have a rod-like secondary structure containing a central conserved region and a terminal conserved region. It is most closely related to citrus viroid V with 56% shared nucleotide identity and is also grouped to the genus Abscaviroid as shown in phylogenetic analysis. It is also stated that DVD is the only viroid known to infect orchids and the third one from monocotyledonous plants. On prions, chronic wasting disease or CWD is a type of TSE that causes a fatal spongy degeneration of the brain. It mainly affects cervid or deer species that leads to emaciation, abnormal behavior, and loss of bodily functions. It is caused by an unknown spectrum of prions and little information on prion evolution is known. Because of this, A study conducted by Velasquez and his team in 2020 aimed to understand the diversification of prions. Cervid species express cellular prion protein molecules with varying amino acid composition. These PRPC polymorphisms can affect prion transmission, disease progression, neuropathology, and the emergence of new prion strains. In their study, they monitored the conformational and phenotypic characteristics of CWD prions, passage through deer and transgenic mice expressing different cervid PRPC polymorphisms. They found out that transmission through hosts with distinct PRPC sequences diversifies the PRPCWD conformations. In addition, stabilization of a new prion strain occurs when it is passage in host environments that restrict prion replication. Results suggested that the diversity of CWD conformers is continuously evolving, wherein CWD prion plasticity and PRPC polymorphisms are significant to prion strain evolution.
There are many detailed publications online that discuss the treatments used to eliminate viroids. Some already proven effective treatments include thermotherapy, cold therapy, and meristem culture, among others. On the other hand, scientists are still working to understand prions and are still searching for an effective treatment. As we already know, prions make use of cells within the body to cause the conversion of a prion protein into a pathological form, which we call PRPSC proteins, or the proteins that cause neurodegenerative disorders. And there's currently yet no cure for these prion diseases. However, the article entitled Autophagy Pathways in the Treatment of Prion Diseases, published in 2019, is one of many other researches that try to find the still undiscovered cure. The journal discusses how autophagy, a cellular degradation and recycling machinery of the body, can be used to fight against the pathological prions. Basically, autophagy delivers a special cargo to lysosomes present in the body, and when the flux of autophagic cells is increased, there is also an increase of PRPSC degradation or PRPSC lysis. As autophagy is the way the body cleans out damaged cells, which is what it would do to the pathological proteins, introducing chemical enhancers for autophagy can improve its therapeutic effect against PRPSC. Using test models, the researchers were able to provide a solid proof of concept for this anti-prion strategy. Now, other than the lysis of prions that autophagy brings, other effects and uses of autophagy in treating prion-caused diseases includes the reduction of exosomal release of prions, which eventually helps in the decrease of prion infections from cell to cell. Moreover, the article discusses how pharmacological induction or laboratory stimulation of autophagy can help in the control of PRPSC, and also may be used for other future therapeutic procedures that may or may not involve prions.